Hello, everyone, and thank you for joining in for Harnessing Invariant Natural Killer T Cells for Off-the-Shelf Cancer Therapy presented by Dr. Lili Yang. Dr. Yang received her PhD degree in biology from Caltech, studying with Dr. David Baltimore. She's currently an associate professor of microbiology, immunology, and molecular genetics at UCLA. Her research covers tumor immunology and cancer immunotherapy, with a special focus on developing gene and cell-based immunotherapy for cancer. So far, her work has impressively resulted in over 60 publications, 20 patents, 2 clinical trials, and 2 biotech startups. For a complete biography of Dr. Yang, you may click her name in the presenter window on your screen. I'm Dennis Chang, Product Manager of mRNA Service. I'll be your moderator for this talk. We encourage you to participate in this session by submitting any questions you may have during the presentation. To do so, simply type them into the field below and click send. You may also submit any technical issues here as well. So with Dr. Yang's video, we had some technical issues. So she'll appear after the presentation for the Q&A. And if you see her screen flashing red, that's not your problem. It's a technical issue from our end. We'll close her camera for now. And without further ado, Dr. Yang, please feel free to start your presentation.
Yes, thank you very much, Bruce, for your very kind introduction. And I thank Joyce and the other organizer, invite me to join your very exciting symposium. Today, I have the pleasure to share with you my lab's research. We work on this interesting type of cell called the invariant natural killer T cell, iNKT cells. I'll give you some introduction about what those cells are and share with you some recent progresses in my lab, like on utilizing those cells for cancer immunotherapy. So what are iNKT cells? If you consider a drop of human blood, it contains about 10 million total blood cells. There's about 0.1 million white blood cells. Those are the immune cells. Okay, and within the immune cells, we have around 5,000 conventional T cells. Those are the T cells we commonly know. And when we talk about the CAR T cell therapy, other cell therapy, those are the T cells we talk about. We have around 10 iNKT cells in one drop of the blood. Very small number, okay? But in the war against cancer, if you consider the 5,000 conventional T cells can form the foot soldiers, really can participating in the big battles. The iNKT cells, despite that small number, they can be considered as special forces. Now in small number, but they move faster and they behave stronger, for joining the war, attacking cancer. So what really those cells are, okay, in scientist's eye? In scientist's eye, they are a small subset of alpha-beta T cells. Unlike the conventional alpha-beta T cells, they recognize the classical MHC1 and 2 and they recognize protein, antigen. iNKT cells display this semi-invariant TCR. So all the iNKT cells within a person and across different people, they share the same T cell receptor, okay? That T cell receptor recognize something MHC1-like, but not classical MHC molecule called CD1D. And that display an antigen, not protein antigen, but the glycolipid antigen. Okay, and those cells, they have both the T cell feature and the natural killer cell feature. They can utilize multiple mechanisms for tumor killing, and they can certainly be engineered to express CAR antigen to enhance and direct tumor killing. And most importantly, because they do not recognize the polymorphic MHC molecules, those cells can be safely transferred across different individuals. So there's no GVHD risk and makes them a very attractive candidate for utilized as allogeneic cell therapy. But there's one challenge, right? So as described, there's very low number of them, in the peripheral blood, and there's high variability between people. So if we try to expand those cells from a healthy donor, peripheral blood, we are talking about a range of 0.001 to 1%, within the blood to be those cells. So make it a very challenging task to expand enough of those cells reliably, for the therapeutical application. So how do we overcome that limitation? There are multiple strategies, people can apply, and the specific strategy my lab has been focused on is to use the stem cell engineering technology. Okay, so we aim to engineer stem cells to differentiate into the special type of very, useful cells. I'll tell you how we do that. Here is our strategy. Okay, so we go to the stem cell called a hematopoietic stem cell, HSC. Another name is called blood stem cell. Those cells live in the bone marrow, of adult human, give rise to all lineage of blood cell, including the all the immune cells, including iNKT cells. Okay, so how do we do that? We utilize a technology, strategy called the T cell receptor, transgenic technology. Okay, this technology depends on what kind of T cell receptor we engineer into the HSC. And then if we program the differentiation properly, then we will, generate the mature T cells. And that will carry the feature of the originality of the T cell receptor. So my own personal research, like back in many years ago, when I was a PhD student, I proved in my research that if we gave the CD8, T cell receptor, we will be able to generate CD8 cytotoxic T cells using the strategy. If we give CD4 helper T cell, receptor, will generate helper T cells. And in my independent lab at UCLA, that we grow, interest in this iNKT cells, and we really like the potential for the allogeneic cell therapy. So we ask the question, if we utilize strategy, if we gave the iNKT T cell receptor to the hematopoietic stem cell, can we program the generation of the special type of cells? And I'm happy to report the answer is yes. Okay, so we published the first proof principle, study in 2015, proof like in the animal, in a mouse model, like we can produce the mouse iNKT cells in this way. And of course, like in order to think about a clinical application, we need to prove the strategy also work for human iNKT cells. And this is the focus of the topic today. I'm going to show you, quite a bit of data like generated from my lab showing the feasibility of this approach. So the work, the initial work has published in 2021, Cell Report Medicine. Those are the two very talented graduate students, Charlie and Alice, who has been leading, that research. So what we have been proved is that we showed the preclinical development of allogeneic HSC engineered iNKT cells for off-the-shelf cancer immunotherapy. We showed that we can generate high number of those with purity and robustness. We showed that the engineered cells can effectively target tumor cells using multiple mechanisms. And we also showed those cells exhibit a high safety profile and a low immunogenicity. Those are very important features to develop allogeneic cell therapy. And overall, that's a set of pretty comprehensive preclinical study demonstrate feasibility, safety, and cancer therapy potential. And the following slides, I will go quite quick and just highlight, some major part of those preclinical data. First is the production. Okay, so here is the approach we use. We use the human CD34 positive HSCs. It can be, either from the cord blood resource or the GCSF mobilized peripheral blood. Okay, so those cells will be engineered with a lentivirus to deliver the necessary transgene, absolutely including the human iNKT T cell receptor. Okay, and then we put it into a in vitro culture. The reported, research, we use this artificial thymic organoid culture, ATO culture, and we do this in collaboration with our colleague Gabe Crooks at UCLA, whose lab developed this very nice, ATO culture, which supports the human T cell differentiation in a culture dish. Okay, and later on, my lab, continued to develop a more advanced version of the culture, and I'm happy to mention. And now we can, grow the cells at similar yield and purity, using a totally feed-free and serum-free culture procedure. Okay, so the take-home message is it's a very robust, procedure. From one single cord blood, we can generate about 10 to the 12th, it's trillion, level of the cells, and this can be formulated if based on the proved CAR T cell therapy. It can be formulated into about 1,000 to 10,000 doses. Okay, and those are really pure. It's almost contain clonal transgenic cells, and the cells, the overall genomic feature looks like very much like their endogenous iNKT counterpart. Next, we look at the phenotype. There are many surface biomarkers we check. Overall, we find that those cells display typical iNKT cell phenotype, which means they express really high level, of the effect molecules and the homing molecule. Those are very useful, for their intention used for cancer therapy. And next, we looked at the tumor targeting capacity of those cells. As mentioned, as this special type of cell, beside the T cell feature, they also have the natural killer cell feature. So this study, overall shows that even without any further CAR engineering and other engineering, those cells can already have an intrinsic capacity to target a quite large range of the tumor, human tumor cell line. Over here listed leukemia, multiple myeloma, melanoma, lung cancer, prostate cancer. So they already have some decent ability to attack those tumor cells. And as mentioned, we can, think about further engineer those cells to further improve their tumor targeting ability by incorporate the tumor targeting molecules such as chimeric antigen receptor CAR, right? So in this case, that after we generate the iNKT cells, we incorporate one more engineering step, using a retrovirus over here, deliver a CAR. Here is a BCMA targeting CAR. So the engineered cells now can target BCMA positive multiple myeloma cells. Okay, we can successfully generate those cells at very high, efficiency, and the ended up results of the cells now have three, mechanisms to attack the tumor cells, right? They can use their intrinsic iNKT T cell receptor. They can use the intrinsic NK targeting, capacity, and they can also use the transgenic CAR, receptor to really guide the recognition of the tumor cells. And all three mechanisms can work together. It also, provide another attractive, feature, which is if a therapeutic cell can target the tumor through multiple angles, multiple mechanisms, then that can dramatically reduce the chance of the tumor to mutate certain antigen and get away from the immune attack, phenomena commonly known as immune evasion. Okay, so we have, tested all the in vitro culture and shows that the cell are very capable and how they behave in vivo, right? So like it's a combination of the tumor attacking ability and the ability to persist and, really traffic to the tumor side within a living creature. And in this case, we use the preclinical mouse model. Okay, it's a mouse model, but we grow human tumor and we transfer the human therapeutical candidate cells in the animal model. Okay, so overall message over here, we tested the low, the heavy load and the relatively lower tumor load, situation, and we showed that at the relatively controllable, tumor load situation, our therapeutical cells, the allogenic BCAR NKT cells, can effectively eliminate tumor, okay, and then achieve this long-term tumor-free survival, okay. And for a very heavy tumor load, like it's possible to eliminate all the tumor, the cell can still effectively control the tumor growth and then significantly elongate the survival of the animals. I should point it out that in all the studies, we include the conventional BCAR T cells as a benchmark control, and compared to the benchmark control, the allogenic, HSC iNKT cells did show, quite a bit of advantage, in particular for the long-term survival of the animal, because of the both the, strong anti-tumor efficacy as well as the free of GVHD, capacity, and that is not the case for the conventional T cells. Yeah, further, on safety, if we trying to develop a novel type of cell therapy product, safety, of course, is, enormously important, right? So two kind of safety, concern over here. Number one is the GVHD, right? We mentioned that fundamentally, those cells should be free of GVHD risk. And we use this classical mixed lymphocyte reaction assay to confirm. When we mix the cells with random donor, PBMC, we don't see any response. Okay, and then that's confirmed in the animal model. Like if we long-termly tracking the animal and look at the individual tissues, we don't see any, sign of the tissue damage and the inflammation. Okay, and again, it's very much different, compared to the conventional T cells, which readily, induce the GVHD and cause massive tissue damage in transplanted animals. Another is general toxicity. We track the animals for a really long time, track the body weight, track the behavior, all of those. And we didn't observe any, obvious like health concern and side effect for the cell receive the allogenic HSC iNKT cells. Another important consideration for developing allogenic cell therapy is those cells need to sustain, persist in a recipient animal with a functional immune system, okay, which means they need to resist to the immune rejection by the host immune cells, and mainly T cells and NK cell mediated immune reaction. So here we have developed, many assays to assess those two features. The take-home message is that overall, those transgenic engineered cells, they express a low level of surface MHC, molecule, and therefore they resist to T cell mediated allo rejection, and meanwhile they also express a low level of surface stress molecule that gives them resistance to the host NK cell mediated allo rejection. So the combination message is that those cells, are safe, GVHD risk free, and potent tumor killers, and meanwhile they show this low immunogenicity and can resist to the allorejection pretty well. And all of those combined together makes them attractive candidate for developing the allogenic cell therapy. And the further, gene editing can be incorporated if we want to further, engineer the cell to make them even lower immunogenicity, right? To eliminate all the surface MHC molecule. That can be done by incorporating CRISPR-Cas9 gene editing step, and that can be achieved at a really high efficiency without interfere with the cell production and the yield and the quality of those cells. And gene-edited cells still can be produced, can be functional, and both in vitro, in vivo assay, confirmed that. Okay, to summarize our ongoing, research, we have successfully developed a feed-free, serum-free culture method to produce the allogenic CAR iNKT cells. There are preclinical studies ongoing to develop the allo-CAR NKT cell therapy for a large array of blood cancers and solid tumors. And the name over here you can read in multiple myeloma, AML, BALL for the blood cancer, and for the solid tumor, the ovarian cancer is a leading target, but we also can cover breast cancer, prostate, pancreatic, kidney, lung, and liver cancers. This technology is licensed by a biotech company, RPA Bio, for commercial development. I'll go a little bit further, like beyond iNKT cells, to tell you a little bit bigger picture what are the other research direction ongoing in my lab. Okay, so beyond the iNKT cell, there are many, turned out to be the many other unconventional T cells, also named innate T cells, have some, interesting feature similar as iNKT cell, but of course have their unique capacity and certainly have a different, aspect of the application for the therapy, even targeting cancer, right? So those are named called gamma delta T cells. They are mucosal associated invariant T cell mate cells. So my lab are working, on those cells too. And currently we are, doing stem cell engineering, focus on the hematopoietic stem cell, but we also want to extend the engineering, go down further, right, to the origin of the cells. So the pluripotent stem cells, like iPSCs, we're developing those technology now. So beyond directly attacking tumor cells, iNKT cell, as well as other, the cells we're working on now. And some of them have this attractive feature of attacking the tumor microenvironment, okay? So besides directly kill tumor, they can also modulate the tumor microenvironment and change the immunosuppressive environment and make this whole, therapy more effective. And that's a feature we're focusing on harnessing, right now. I'll stop over here and thank my lab members, especially the one highlighted in red over here, those students, current member and past member participate in the work I just showed you. I would like to thank our collaborators and the funding support. And importantly, I would like to thank you, audience, for your attention. I'll be happy to answer any questions you may have. Thank you.
Hey, thank you, Dr. Yang, for your excellent presentation, and really excited to see your result and the future applications of it. And we'll now start the live Q&A session of the webinar. And if you have any questions you'd like to ask, again, please feel free to put down your questions on the chat into the chat box below. So let's start. I see we do have some questions. So the first question, Dr. Yang, could you comment on the potential of iNKT cells for treating solid tumors?
Certainly, yeah, Bruce, this is one, aspect actually we really like, interested about iNKT cells. So as mentioned, the iNKT cell, they have this special T cell receptor, and recognize the CD1D, molecule, right? And presenting the lipid antigen. So there's something special about the CD1D and the lipid antigen in solid tumor microenvironment. When we consider its immunosuppressive tumor microenvironment, they are composed by the immunosuppressive cells, right? The most studied include the tumor-associated macrophages and the myeloid-derived suppressive cells, MDSCs. And those two types of cells, interestingly, happen to express a really high level of CD1D. Okay, and the solid tumor microenvironment happen to contain really high level of the lipid antigen that can bind to CD1D. So a combination of that is that a big part of the immunosuppressive tumor microenvironment in solid tumors are natural target for iNKT cells. So we are very actively pursuing that and actually see some quite dramatic results. For instance, if we collect the primary solid tumor samples from cancer patient, if we mix those samples together with our engineered iNKT cells, we can see very effective and very specific elimination of the TAMs, MDSCs. And that happened to not only one type of solid tumor, it happened to almost all the solid tumor type. We tested more than 5, okay? So like tumor types. So we think that can be a very attractive, unique aspect of the iNKT cell-based cell therapy compared with the conventional T-cell therapy, because conventional T-cell therapy are not equipped with that part of the function, right?
Thank you, Dr. Yang, with the explanation. I think following on that, we do see some more questions. So here we have another audience asking, what are some obstacles or limitations that remain to facilitate or improve the manufacturing of iNKT cells for therapies?
Certainly, yeah. So, for the manufacturing, is that how to scale up, right? And how to, have the CMC procedure really robust and low cost, right? So like all of this, it's a new cell therapy platform. I would say it will be, the first in-human kind of trial, we are doing. So there's no very obvious obstacle. Okay, HSC culture, collection, especially from the cord blood origin, the supply chain is established. The culture protocol is pretty much established. We use a lenti-vector, based gene delivery, and that's pretty well established, and especially in the context of engineering HSCs. Okay, and the expansion protocol, of course, is the new, technology, we developed, and it's a feed-free, it's a serum-free. So like fundamentally, we don't see too much problem for scale up, and in practical, it still needs to work out. I think that is the commercial side, the startup company Apia Bio is focusing on, doing this.
Yes, yeah, that makes a lot of sense. Yeah, I think scaling up is always a problem, but it seems like for this solution, we have less limitations, and we're facing less challenge with this as well.
Yeah.
I think, okay. So I think we do have another question coming up. So this question was around how do you engineer iNKT cell differ from conventional CAR T cells in persistence and exhaustion properties?
That's a very good question too. Like all T cells, those they are not like out of the box and unnatural cells. Okay, they are T cells. They certainly will share the machinery, like control the exhaustion, control the, dysfunction, of the T cells. So when they encounter tumor and hang around in the tumor, not too much. Okay, I would not say iNKTs are just uniquely, immune of that. They're not. But we can think about, engineering strategies to help them, to overcome it. And we can certainly improve the culture condition to make them more fit, right? So the fact that the iNKT cells we study over here derived from stem cell instead of just enormously expanded from the mature, cell pool from the peripheral blood make a difference, because coming from stem cell means they are more like at the younger, age of their life. Okay, the telomere will be long, kind of the immune potential, the memory potential will be higher. Okay, that's number one. Number two is that the engineering approach you can certainly think about, right? So there are some, molecules already known. It's very helpful, for improved persistence and the memory, formation and maintenance of the T cells, and which work for iNKT cells, and also work for the other immune cells. One of such molecule, for instance, is IL-15. Okay, so incorporate IL-15 transgene, which we actually are doing now in our research, can really help, to further improve the in vivo performance. Okay.
Okay. Thank you, Dr. Yang, for walking through the different differences between iNKT cells and conventional CAR T cells. So I think that was very clear. And let's see if we have any additional questions coming up. So if you have any further questions, just a reminder, please put it down in the chat box, or after the talk, you can also reach out to Dr. Yang through our channel. Okay, so I think we do have 3 minutes more. If there's no more questions, I think thank you very much again, Dr. Yang, for today's talk. And it was very exciting to learn about your research, and really looking forward to its future applications. It was very good. Thank you again for attending today's session, and also the audience as well. Thank you very much.
Thank you, Bruce. My pleasure. Bye.